Hello everyone. So today I am going to present on the topic AI driven target characterization for structure based drug design. So this topic mainly consists of AI driven approach for structure based drug designing. And for this presentation I will work on predicting the structure of a renowned enzyme called isocitrate lyase, as you can see on the screen. The enzyme for our today's topic is isocitrate lyase, which is a lyase classified enzyme which produced which is produced by Mycobacterium tuberculosis when they are present inside the latent TB, inside the body cells during latent TB. And this lyase, isocitrate lyase, helps in converting isocitrate into glyoxylate and succinate, and takes part in glyoxylate shunt, which helps the bacteria to survive inside the body cells during latent TB. We will be following these steps in this video. First, we will predict our protein structure using AlphaFold server. Then we will validate this protein structure using PyMol and by comparing it with the PDB structure already available, and make suitable changes to it according to our docking protocol. And then we will do structural validation using Ramachandran plot. And then if found that energy minimization needs to be done, then energy minimization will be done using Swiss PDB software. Then DeepSight AI will be used for predicting binding pockets of this energy minimized protein structure. I will show you the steps in the coming videos. Doing our work, we'll first visit Unicode. Here, we'll first write our Unicode ID P9WKK7. As you can see, this is isocitrate lyase. Sequence 428. Okay, ICL created by ICL chain. So first we'll go to the first sequence here. If you see this website, here in your left side there will be functions, names and taxonomy, subcellular location, phenotypes, variants, PTM or processing expression, interaction, structure, family domain, sequence, like this. From here go to sequence. This is the first sequence. You need to copy it. After copying it, go to AlphaFold server. Using AlphaFold server, we will build our protein from the sequence. We need to first clear it. Then we need to first put our sequence. We have already copied it from UniProt. Now we will paste it here. Copy sequence is 1. If you want, you can give the copy sequence as 2 also. It is not a problem. But if you go for 3, if you go more than 2, it will it will take up to 2 hours, which is quite a large amount of time. So we'll go with 2 hours. See, if we are going for 2, it is not giving any a number of time amount of time needed. So we'll go with first we'll go for a trial session. We'll go with 1 second, 1 copy only. Continue and preview job. Job, right? Job name. I will write my job name as ICL YouTube video or ICL video. Okay. Confirm and submit job. After extracting the zip folder containing the AlphaFold results, you will get this many results. From here, directly go to summaries, summary folders, sorry, summary files. See you need to find the files called fold with your protein name video, like sorry, fold with your protein name, then summary, then confidences. Confidences 1, confidences 0, confidences 2, confidences 3, and confidences 4. This total 5 video, sorry, total 5 files will be there. From here, go for confidence 0. Notepad, open the video. Sorry, open the file. You will see that already here is written this last 4 values: fraction disordered 0, hash class 0, num cycles 10, ptm is 0.98, and ranking score is 0.98. This ranking score shows that our protein is highly our protein is predicted from the sequence with high confidence, and it is quite close to the real world protein structure with one sequence only. So after opening the summary files in Notepad and seeing the confidence score, by seeing the ranking score and uh, by seeing the ranking score, you will need to open CIF files. So going to CIF files, you will see that there will be some files known as template. Don't click it, because the template files are the files which AlphaFold has used for predicting our protein structure. It is not needed by us for our further predictions. But you will need to see this fold with hyphen, then our job name, and then model 0. Model 0. You need to open model 0, but you need to open model 0 in Pymol software. So I will open the Pymol software in my computer. If it is there in your computer, it's quite good. Go to Pymol. So here I will first open Pymol. In Pymol, I will open my file summary, not the summary one. Sorry for the mistake. It will be CIF. From CIF, go to this model 0.cif. See, it will be this. So you need to convert this CIF into PDB structure. So for converting CIF into PDB structure, go to files. So go to file in the top panel, give it in Pymol. In Pymol, in the top panel, you will see file, edit, build, movie, display, setting, scenes, mouse, wizard, plugin, and help written. So go to files. If you go to files, this options will come in your screen. From here, go to export structure. From export structure, go to export molecule. Export molecule, go to save. Here, since you need to download it as a, if you want, you can download it in the same folder also. But you need to change this to PDB, because I prefer mostly PDB only. So PDB from PDBX. So here I will give it a different name, ICL, not ICL video one. It is first I have made it with that only. So now we will save it a different name. ICL uh, YouTube. ICL YouTube. So I will save it. It will be saved as a PDB folder. So PDB file. Then there is no need for now to open this file in to open this file in Pymon. Now we can go out. Now we need to open Discovery Studio. After going to Discovery Studio, you go to the in the Discovery Studio. It will be given a file, view, chemistry, structure, sequence, chart, scripts, use Windows help. go to file directly, and this please download this Discovery Studio file in your computer. So Discovery Studio software in your computer. So because the Discovery Studio quite helpful in for doing post-docking analysis also. So after docking, you need to go to do the post-docking analysis. So you need Discovery Studio for that. So please download, open Discovery Studio. I see YouTube. See this file. In Discovery Studio, you need to give CTRL plus H. If you give CTRL plus H, you will see panels opening here. So here it will you will see protein groups and chain A, because we have predicted, if you remember, we have predicted only one copy number as chain. So there will be only one chain here. Although we'll now see it in PDB how many chains it has. Since I already know the PDB, but okay, I will see you from the see show it to you from the beginning only. After viewing our protein in our predicted protein in Discovery Studio, we will see the same protein in PDB, protein data bank. So for this, go to Chrome and write RCSB space PDB and see that there is written RCSB PDB database. So you just need to click RCSB PDB, go to the home page of RCSB PDB. Since I worked with this protein and I know the PDB ID mostly cited in literature, so I will give the PDB ID. I will go for the PDB ID 1F8M. Okay. So since we are working on isocitrate lyase, as I have taken its sequence or the sequence of that protein only from UniProt, so I will check for isocitrate lyase only, whose PDB ID most cited in literature is 148M. See, if I search for one F8M, you will see that here it is written crystal structure of 3-bromopyruvate modified isocitrate lyase from Mycobacterium tuberculosis. So it is isocitrate lyase only, but it will have an inhibitor called 3-bromopyruvate or pyruvic acid. This same pyruvic acid will be there as a ligand here, or inhibitor here. So we'll then see the method used for the protein uh, protein for weight level structure prediction of protein. This protein the X-ray diffraction resolution is 1.80 angstrom. So mostly for if you go for PDB, PDB structure, try to take PDB structure whose resolution is between 1.5 angstrom to 2 angstrom. 2 angstrom. So since it's 1.80, it is good, like medium. It is medium. Medium resolution is there. If it is high resolution means that you will see the structure clearly in PyMol. But anyway, we will go for this structure only since it is highly literature cited. So if you see here, this protein have in according to PDB, it has four chains A, B, C, and D. And in the Discovery Studio, I have already shown you that our protein have only A, right? So its sequence length of 429. So then we'll go to below. There is, if you can see here, two ligands are there. One is pyruvic acid and another ions are there. Ions are there present in the in this protein, magnesium ions. But according to AlphaFold structure predicted, there are no magnesium ions, right? See, there is only chain A and protein group. So there are no magnesium ions present in our structure. So we need to keep this magnesium ion. We need to keep this magnesium ion like the PDB, like the PDB ID, like the structure available in PDB. This 148M. So we need to add magnesium ion as available and at the position available in this PDB file. But we don't need this pyruvic acid because it is an inhibitor. It is not needed. During docking we need to cancel this, remove this inhibitor only. So we are not adding this for our um, in our alpha-fold structure. So we'll go with this magnesium ion only. We'll need to add this magnesium ion. So please download this magnesium ion. So for downloading any structure from PDB, go to download files. From there, go to legacy PDB format. Here, legacy PDB format, legacy, but I usually prefer legacy PDB format only. So I will go for legacy PDB format. So you can see 1F8M since I have downloaded many times. So it is just two. It is same copy only. So 1F8M. So I will open this 1F8M to in the Pymol, and we will align them. We'll align our predicted structure and we'll align this protein, this PDB ID structure. So we'll again open this Pymol, but first we need to cancel the Discovery Studio tag because computer becomes slow if you work on many softwares at once. So after canceling Discovery Studio, we'll again go to Pymol. So please take up notes from here. So in Pymol, will give particular commands for aligning the protein, aligning the two proteins, that is our protein and then this our predicted protein and PDB ID, PDB protein, PDB structure available. So for adding adding our structure, our predicted structure in Pymol, again go to file. We will add the two structure only. So first we will go to our first we will open our structure. Okay. So we'll go to files to open. Then we'll go to this downloads. We will go to ICL YouTube if you remember properly. This is the PDB ID we had created from Pymol earlier, right? So this one, and then we will also open PDB file. PDB file we just downloaded 1FM82, right? So if you see properly, structure is quite big and complex. But since we are working on only one chain, so we will first cancel out the other chains. Okay, in 1F8M, uh, but for this we will cancel out only chain B, C, and D. Because if you remember, this protein have four chains, and this protein is a homotetramer. So A, B, C, and D, these four proteins have four chains are there, and all are similar chains. So we will cancel B, C, and D. We'll keep only A. Okay. So for removing the proteins, first you need to see the programs clearly. Comments clearly I am giving. First give remove 1F capital F 1F 8M2, right? Because this is 2. So and space and I think if I magnify, yes, okay. Can you see the below screen properly? So give this command 1F8M like your PDB ID you have taken from PDB RCC PDB. Remove your PDB ID and I will give a not chain A. This command, what this command will do, this command will cancel out chains B, C, and D, but not protein chain A. I think some problem is there with this, so I will do it again. No issues. Because if you see properly, it is written, it has some error is there, right? Invalid selection 2, right? It is given. So if you see properly here, it is given 1F8M hyphen 2. So I need to give it like that. So again, I will write it, remove. This time I will give as given in Python 1F8M hyphen 2. Then and I will write and not chain A. So what this command will do, it will cancel out all the other chains in the PDB. in the PDB file. I have opened two files in this workspace, right? One is our predicted protein and one is this complex PDB ID protein. So now I'll give it again. See, now if you see properly, you can see that only one chain is there. This chain is only there. If you want to see it properly, see, like these two chains are there. Only these two chains are there. So this green one is our protein. This green protein is our protein, like our predicted protein, and this blue one, with a single chain and very large amount of water molecules around there, with red dots, which shows water molecules. So now you need to align this to a structure. So for aligning this to a structure, give this command: align our AlphaFold model. Our AlphaFold model is ICL hyphen C in Pymol as it is given there, ICL YouTube, comma, give a comma, then reference PDB ID. So reference PDB ID is 1F8M hyphen 2. See, see now if you look closely, there are two proteins, our predicted protein and our AlphaFold predicted protein and this uh, PDB taken structure protein, both are aligned in a single structure, right? So now from here, from here you need to remember that you need this magnesium ion there only, right? If you remember it properly, this pyruvic acid is not needed, okay? But you need this magnesium ion to be present because magnesium ion acts as a cofactor for isocitrate lyase according to literature, and it is present in active site of the protein. So you need to have this magnesium ion present in during the docking. So now you need to find the magnesium ion, right? So give this command: select mag, right, as it is given, mag as I am giving it, write it. Select mag hyphen ion, comma, resn mg, because according to Pymol they gives capital M and G, capital Mg for magnesium ion. So you will find this new thing created there. Okay, so first you will get selected, magnesium ion will get selected. Then go to then you need to see the properly the magnesium ion by zoom, zoom, then space mag space ion. Okay, you will see this magnesium ion is always zoomed, and you can see this around red dots will be there. These are all the water molecules, and if you see there is a green color also, this is another inhibitor there. So you need to go with this magnesium ion only. You don't need this other ions, other inhibitors ions. So what you will do, you will then you need to view the magnesium ion as sphere. So write this command: show sphere, show space spheres, then space mag ion. I think you can show below my screen. I am writing the command mag hyphen ion. So this is the magnesium ion, this one only. So okay. So after this, after this you need to make it color yellow. Okay. So go to this, write this command: color space yellow, comma, mag, then hyphen ion. See, the color changed to magnesium ion. So you are quite confirmed now that this is the magnesium ion. Okay. So this is the magnesium ion. Okay. So now what you will do, you will hide the non-bonded because if you see properly, there is red dots here, right? There are some red cross. There are some red links there, red cross-like structures there. So you need to delete them. So what? How will you do them? You hide non-bonded, write it as once, non-bonded, comma, 1F8M hyphen 2, 1F8M hyphen 2. See, all those red color crosslinks are over. They are not there now, right? So those things they are present before. See, these red colored crosslinks are there, like crosses are there. So those are all the non-bonded atoms. Those atoms are not bonded to the protein structure in PDB, right? So now you can now after giving the comment, it is not there, right? So now you go to this zoom mag. It will be same program only, so not an issue. So after this, I think you can see the screen properly. So turn off the light against 1F8M2 or your protein, whatever you are working on. Since we have aligned this and then um, uh, remove the non-bonded ions, sorry, non-bonded atoms. So now since we have also turned off the our PDB from this bimol, so now we will have only our AlphaFold predicted model and magnesium ion. So now you need to create is a single object. So our single object name will be final target or final. It is final is fine. Create final, comma, then our AlphaFold protein model name that is ICL YouTube, ICL hyphen YouTube or magnesium ion. And for primal, the magnesium ion is written as MAGION. So I think you can see now the protein. See the magnesium ion is turned as yellow here. I think you can see it here. Yellow is there. So it is present. It is an active site. It is present in one of the active site of this protein, isocitrate lyase. Similar to this, since we are working on only chain A, similar to this chain B, C, and D will also be there. But since we are predicted only one chain, so like this. So now we can continue the work with file. From file, open the file again in Pymol and go to this export structure. In export structure, go to export molecule. From export molecule, give not give enable, give final because the final target consists of those two, right? Final target consists of our alpha fold model and magnesium ion, right? So save it. Save this as final ICL, sorry, save this as ICL final. And this is our final PDB. But till now we'll not confirm it. We have some other works also with it, right? So go to PDB because I prefer PDB more. PDB, then save. Okay. So our work is done here, but now we need to see it in Discovery Studio to verify whether magnesium ion is added to our protein, to our alpha protein or not. So go to Discovery Studio again. It is opening. From there, go to file. Top panel where they are containing a file, edit, view, chemistry, structure, sequence, chart, scripts, tools, Windows, help. Go to file. They are open. ICL YouTube, right? It is not created now. It will be there again. We just now created this file, right? It is not the same file. I will first search the file. ICL final, right? This one. We'll open it again in Discovery Studio. You will again go to mouse mode, more mouse mode, go to rotate. Is it not there? We'll check it once. See, it is there. So we again give CTRL-H. Okay, we'll open it again. ICL final. Then we'll give CTRL plus H, that's Control plus H. See here, heteroatoms will be there. To check which heteroatoms are there, just go to this side panel. If you can see, uh, their heteroatoms is written, right? So check. See, first one is only the heteroatom. Mg of 451. This one. This heteroatom is there. So if now you know that here magnesium ion is there already in our file, right? So till now we have predicted the protein structure using AlphaFold. Then we have aligned it with the PDB structure using Pymol, and then added the magnesium ion as available in the PDB ID to the to our AlphaFold structure at the same positions as available in the PDB. And now and now we need to validate the protein structure. For validating the protein structure, we'll need to do Ramachandran plot analysis. For doing Ramachandran plot analysis, we have to visit this website, SAVES 6.1. Okay, so as you can see, this website's name is saves version 6.1 structure validation server. So this server is used to validate the protein structure predicted using using homology modeling, or using ab initio, or the other methods. So we need to validate our protein structure we have already created and seen in Discovery Studio earlier. So we'll first visit this website. As you can see, this is under UCLA. We will choose our file, which is ICL final. Now I will run the programs. Here you can see that various options given: ERAT, verify 3D, proof, what check, pro check, and open. So pro check is the option we need to go for Ramachandran plot analysis. Okay, but before that we will press ERAT also, and we'll go for pro check also. So it will take some time. See, if you see here, overall quality score is 9.7981. So this project will take some time to go for. I will show you the direct results after they publish it. After running a Ramachandran plot analysis in project uh, via Save 6.1, finally got the result after 20 minutes. As I click the results are being loaded. It will take some time. So this is the result of the Ramachandran plot as we ran it in Save's website. As you can see, there are following options here: Ramachandran plot, all Ramachandrans, Chi, two plots. So from this we will go with for Ramachandran plot. Okay. See, this is the Ramachandran plot. From here, if you need to just see this result: residues in most favored region. As you see, this residues in most favored region is 356. In in additional allowed region is 22, and the generously allowed region is 2. Please remember it properly. So now we will see the PDF file for this. See, this is the result of the Ramachandran plot, and we need to see this result only. And if you see properly, there are residues in disallowed region as 0. So in a way, this protein is quite okay to go for. And also most favored region residues are 93.7%, and allowed additional allowed region is 5.8%. So now we have to do energy minimization. For doing energy minimization, I will use Swiss PDB app. So first I will search for Swiss PDB. Swiss PDB viewer. Actually in my computer Swiss PDB is there. So I will just go for Swiss PDB. See, since I have already downloaded Swiss PDB, it is there. SPDEV4. So here I will go. From here I will go with this. This is the Swiss PDB. So it will open, and then here I will do the energy minimization. Can cancel this. From here. Here if you see properly top panel of Swiss PDB viewer, file, edit, select, build, then tools, feed, display, color, these options are there. So go to files, open PDB file. From open PDB file, we need to select our own model, that is ICL final, as we are working on it. Here we'll go for open. We'll open it in Swiss PDB viewer. There is no point for this. Here, this is this protein as we viewed. This is our protein, predicted isophosphoid protein with the magnesium ion. Here we need to do energy minimization. Our main objective here is doing energy minimization of our protein structure after adding this. So we'll go to tools. From this top panel, we'll go to the tools option. From here, we'll go for energy minimization. I think you guys can see energy minimization is there. CTRL plus N, Control plus N. Energy minimization. Okay, no residue selectors. First, you have to select. Select. From select, go to all. Okay. Then tools. From tools, go to energy minimization. See, progress is it is going on. Progress is going on. See, some of the proteins, these red ones, they have been energy have been minimized for them. So we'll go at the below. We'll go to the below. Okay. Now we need to save this structure as our new structure, and then we'll do Ramachandran plot analysis of this structure again, and then we need to compare the before energy minimized structures, what was the result, and after. So we'll go to save. From save, go to current layer. Current layer, go to not final. This one will be ICL energy minimized. So it is energy minimized video dot pdb. So the structure has been saved. Now I will again go to Save's website and do Ramachandran plot analysis. The same steps I will repeat, and then we will find the difference between before and after energy minimization. Saves again. Save 6.1. Here structure validation server. We will go again. Here we will choose the file as ICL energy minimized video. This one, and we will open it, and then we'll run program again. This in this run program we will do erat. Here it is started and we'll start check. Again the same process will go on. So I'll wait for some time. As you can see, the ERAT is quite good because previously, if you remember, our ERAT was 98 point something, but now it is 99.04. It is quite good. Overall quality factor is quite good. So after doing this project analysis, we can say how much energy have been after energy minimization, how much Ramachandran plot is allowing or disallowing. We can see by comparing with the previous one. Let's wait. Took me around 30 minutes to do the Ramachandran plot for this energy minimized structure. Now we will view the results for this Ramachandran plot. It is loading, the results are loading, and after the results are loaded, I will show you the results. So it means that our structure is completely stable, and now it is ready for docking. But before docking, we will need to see the grid box. Like we'll need to see the predict the active sites of the protein. And for predicting the active sites of the protein, for for AI assisted drug designing, we will need a particular tool known as DeepSight AI Play Molecule. So if you search by DeepSight AI Play Molecule, you will get to see the results. Go to this website open.playmolecule.org, and if you open it, you will see. So this is a particular chat box which is used to predict the AI, which is used in AI assisted drug designing workflow and is used to predict the active sites of the protein. So here we'll directly upload our protein file, which was ICL final energy minimized, ICL energy minimized video. This one, open. From here we will go. From here we will tell this AI to predict. Please predict active sites or binding pockets. The same. Please predict the binding pockets of this protein molecule. Also mention the amino acids present in the active sites, present in each of the active sites, present in each of the active sites. Also give the pocket score. Also give the score of each, the score of each binding pockets. Based on that score and based on literature review, you need to proceed forward in your work for AI assisted drug designing. So after uploading it and giving the proper prompt, the AI will start doing its work, and after, and it will show the binding score, binding pockets, that is active sites, along with the amino acids present in that active sites, yeah, and also the binding score, like the score of each binding pockets or active sites. In this workplace given as black, you will see the molecule also after the binding pocket is predicted in your left side. Active sites of the protein has been predicted. And as you see, deep site are predicted in total three active sites in the protein. Their center as well as residues in the binding pocket is written in this table, as well as their binding score is also given as CSV file. If you predict your protein's binding site using deep site AI, the result uh, the result will be in the form of this table only. And if you want to see the protein properly, see, I think you can see that in the left side of my screen, I am rotating a protein and orange lines are available in that part. So this orange lines means binding pockets region, binding pocket region. The binding pocket region as well as their scores and their centers are given in the right hand side now. So you can use these binding site centers for your docking, and which binding site you will go for, you will select will depend on the scores on of the binding pockets. So so these centers you will be using these centers for the docking as grid box centers.